Good morning, good afternoon, and good evening, everyone. And welcome to the scientist.com and Lonza webinar titled Arrayed CRISPR Screening for Phenotypic Discovery at the Functional Genomics Screening Laboratory, or FGSL. I'm Liam Sanyo from the events team here at scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Ulrike Künzel and Dr. Chun Hao Wong from AstraZeneca and University of Cambridge, respectively, who will showcase arrayed CRISPR screens and discuss collaboration opportunities with the MRC-AstraZeneca-University of Cambridge Joint Functional Genomics Screening Laboratory, or FGSL. And with any further ado, I'm very pleased to welcome Dr. Uli and Wong. All right, I think Wong is first up, but yeah, feel free to take it away whenever you're ready.
Great, thank you so much for joining us today. So we'll tell you a bit more about this new lab that we're setting up that's called the Functional Genomics Screening Laboratory. In today's webinar, we'll go through, in general, what CRISPR is, what we're trying to do here at the FGSL. We'll also present two case studies, one on array CRISPR screening facilitates dissection of complex phenotype, and second, overcoming challenges with CRISPR screening in complex models. And finally, we show a case, a few routes to collaborate with us. So just very briefly, what is functional genomics? Is the ability to associate genes with phenotypes by systematically manipulating the genomes. So there are various tools available nowadays, from zinc finger nuclease, TALEN's, RNAi to today CRISPR. We'll be focusing on CRISPR today, and in the past 10 years or so, there's been a rapid progress building on CRISPR capabilities to really increase the scalability, specificity, as well as multiplexability of tools, and to integrate this with technological advances to really understand human genetics and phenotypes. So for today's webinar, I'll be focusing on Cas9, SB-Cas9 endonuclease. It remains the most widely used gene editor. It's due to its high activity and specificity. It's highly programmable and specific through co-expression of Cas9 as a protein and a short targeting guide RNA that guides the Cas9 protein to the target DNA sequence. It generates kind of a molecular Caesar, if you like, generate a break in the DNA, and it causes double-strand breaks and trigger the repair mechanism, which is frequently quite error-prone. So this would generate gene disruption or loss of function to generation of deletions or insertion within the DNA itself. So CRISPR-Cas9 loss of function screens, and in general, you have the two key components, which is Cas9 protein and guide RNA library. And typically when people talk about CRISPR screens, they refer to a pool screening methods where you introduce your library of guide RNA in a lentiviral format. So you can actually title them to each cell actually receive a different guide in a pool setting. So you can really scale it up and study endpoints such as variabilities or using marker expression using in combination with flow. Typically, they rely on next-generation sequencing for deconvolution, and this is conventionally applied in dropout and expression-based screens, as you can see here with the list of different types of applications that's well known, typically in oncology screens. And I did a recent PubMed search on CRISPR screen recently, and there's more than 10,000 publications around CRISPR screens, really signifying the kind of application of CRISPR screen in identifying new targets per se. I just want to introduce array screens, which is a slightly different kind of format in comparison to a pool screen. So here you actually introduce your guides in a plate format. So you actually have a physical separation of the guides itself within a plate. So plate location 1 could be just one guide, and then plate location B can be a different guide. So you can actually do quite complex phenotyping, including such as spatial imaging, looking at morphology of the cell, cell-to-cell interactions, as well as really combining different endpoints together to have a multi-parametric analysis of quite complex phenotypic endpoints, which we believe is a quite straightforward genotype-phenotype association that allows us to study quite complex phenotype in complex cell model. So target identification through array CRISPR screen, typically you have a biologically relevant model. This could be a disease-relevant model. You have a plate-based phenotypic assays to read out for your functional readout per se. You have a CRISPR-Cas9 reagent delivery strategy that allows you to do interrogation of genes in your cell model at scale. This is typically followed up by a target validation strategy, either through orthogonal assays, integration with other data sets, such as genomics. And this would allow us to generate new hypotheses, understand new pathways on your models, also inform on therapeutic targets, as well as patient stratification strategies. There are various components in an array CRISPR screens, so I just want to quickly go through this. You really start from a library, so in an array format, typically this is a synthetic guide RNA library because lentiviral library seems it's a bit challenging to make. You can go into a genome or druggable or a targeted panel. This is combined with your biological relevant models, can be cell lines, primary cells, iPS, core cultures, and organoids. Combined with your reagent delivery for CRISPR, this can be a lipid-based transfection or electroporation of Cas9 and guide RNA in a complex that we will kind of talk a bit more later. Your assay, which is your functional readout, this could be an imaging endpoints, this could be a flow assay, or any type of microplate assays such as ELISA. We really need to scale this up in order to actually perturb the genome at scale. So this is usually, we usually develop a complete workflow to integrate automated liquid handlers as well as robotic platform to allow us to do this at scale quite robustly. Finally, we would perform analysis to identify hits as well as biological interpretation. So here at the Functional Genomics Screening Laboratories, this is, I just want to quickly tell you a bit more about this new lab. This is an academic industry partnership to accelerate target discovery. We aim to identify new genes and biology implicated in tissue development and disease etiology. I tell you a lot about different type of CRISPR and CRISPR format, but I really want to focus on array CRISPR screening because that's what we're focusing on. We are based in this building that's called that's in the Cambridge Biomedical campus, and the new lab is hosted at the Milner Therapeutics Institute. This is a collaboration between us, the Milner AstraZeneca, and the Medical Research Council at the UK. We focus on complex cell models outside of oncology, and we are part of the UK Human Functional Genomics Initiative. That is a new initiative to facilitate translation of innovative and collaborative research. We have a QR code here for the website as well. I think the link is also in your handouts that you feel free to visit. So our approach for the lab is really focusing on a few pillars, from CRISPR in complex cell models, from primary cells, iPS models, organoids, to functional readouts, imaging, flow, microplate assays, but also developing automated workflows that allows us to do high-throughput screening, incorporation of robotic liquid handling, and automated endpoint platform. We run all our projects through collaboration, either with academic groups or with small, medium-sized enterprises. And the ultimate aim is to advance mechanistic understanding underlying health and disease to facilitate this discovery of novel biomarkers and drug targets. So gene editing in human cell models, as I mentioned, these are the models that we focus on, and we focus on non-viral CRISPR editing, either through transfection of synthetic guide-RNAs in engineered Cas9 expressing models, transfection of synthetic guides and Cas9 in an mRNA format that I will describe a little bit later, as well as electroporation of guide-RNA, Cas9 protein, ribonucleoprotein into the cells that we will present in case study 2. So first case study, so this is to identify regulators of intracellular trafficking and spatial organization. So the background for this is the microtubule motor protein dynein actually plays an important role in cellular organization and cargo trafficking, as we are looking for trafficking and organization within the cells. We use imaging as a readout, which is why we pick array screening format. We develop a high-throughput workflow for sequential mRNA Cas9 lipoection delivery that allows us to have a huge population of Cas9 expressing cells, and then we sequentially transfect them with synthetic guide RNAs in a plate format, and this allows us to do high-throughput imaging. Just a proof of principle with the phenotypic readout. So this is a control where you have the GFP and the RFP, which is a dynein reporter, being quite clustered in the microtubule organization center, which means that you have a normal function of dynein. When we perturb a core regulator of dynein, you start to see dispersion within the cellular within the cytoplasm. This is true as well as if you perturb the core component of dynein itself. So this is quite quantitative that we can measure it. We also develop a multiplex imaging workflow for dynein cargoes at subcellular resolution. We can also capture endogenous dynein dynein cargo, which is early endosomes, using antibodies immunofluorescence approach. So you can see that we can quite nicely multiplexing these endpoints together within one screen. Finally, I just want to quickly show you how the data looks like. So really, it's quite complex phenotype when we are looking for something that one spot, multiple spots across the cytoplasm. It can also be a different distribution within the cytoplasm itself. You can see that this is the, we use this as a normal neutral control and a positive control. So you want to find hits that either behave quite similarly as a perturb control. So you can see a separation between the control here, and we're looking for genes that, when perturbed, fall into this category per se. I mentioned a different screen, which is different endpoint within the same screen, looking at endogenous endosomes screen. Here, we not only that we can look for how they are located, but we can also look for the morphology, how they're different. And I think I just want to quickly summarize this part. So CRISPR-Cas9 screens as an unbiased experimental approach to really identify novel biological targets, and array screens really enable us to multiplex different endpoints, facilitate dissection of complex phenotypes through morphological profiling, imaging in cellular models that I just showcased. For any more details, you can read the recent publication, and I'll pass the presentation to Uli.
Thank you. So now that you've heard about the power of arrayed CRISPR screens, I want to bring you back to our 4 pillars or cornerstones of the FGSL. We want to perform CRISPR editing in complex models using functional readouts and automation, and ultimately work under the umbrella of collaboration to identify new pathways in health and disease, and ultimately identify novel drug targets. Let's focus on more detail about how we want to operate in the FGSL and how we want to perform CRISPR edits in complex models. So in the FGSL, we aim to work with complex models such as primary cells, organoids, and so on, because we believe that they are really valuable because they best recapitulate what the patient or in vivo scenario looks like. So any targets or pathways we identify in complex models are more translatable into the patient. However, these complex models come with challenges for CRISPR screening. First of all, often cell numbers are really limited with primary cells. That limits us with the scale of the screen that we can perform. Whole genome CRISPR screens are often not possible. Second, these models are often very difficult to transfect with conventional lipofectamine-based transfection. So we need to find other methods to deliver the CRISPR-Cas9 reagents into those cells. And third, complex phenotypes are often observed in those models, so it's really hard for a CRISPR screen to differentiate different phenotypic changes and quantify them appropriately. So we need the right tools to do so. I want to present to you some strategies how we can overcome those challenges and move into a case study where we applied those 3 strategies. So first of all, we can reduce the size of CRISPR screens by using bespoke gene lists. Those gene lists can be derived from knowledge graphs or omics approaches, such as transcriptomics and proteomics analysis of the disease-relevant model. These approaches help us to have a bespoke disease-relevant gene list that we want to test, rather than an unbiased whole genome approach, which of course would be desirable, but often not feasible with the limited cell numbers we have in primary or complex models. Second, electroporation is really a beneficial and efficient method for delivering CRISPR reagents into primary cell models. Electroporation can be coupled with semi-automated workflows, which makes it amenable for high-throughput assays and CRISPR screening. Thirdly, to dissect complex phenotypes, we can use AI and machine learning-based image analysis tools. We can use those tools to classify and train an algorithm to classify different phenotypic features that we see in cells, and this can really help with dissecting even small changes or really multi-parametric changes that are hard to quantify with conventional intensity analysis. I want to show you in this case study here how we applied all those 3 methods or strategies and worked with a challenging model. So the case study that I'm presenting was an arrayed CRISPR screen in primary renal cells, where we wanted to discover new targets for chronic kidney disease. The work was performed at AstraZeneca, but I'm presenting it here because this is the type of work that we also will do in the functional genomics screening lab in terms of complexity and challenges that we need to address. So in this particular project, we wanted to understand key pathways in chronic kidney disease and identify novel therapeutic targets. To achieve this, a knowledge graph-derived gene list was predicted using AI, and these in silico predictions should be confirmed or validated in an in vitro model. And for that reason, we performed arrayed CRISPR screens in a disease-relevant cell type. To show you the workflow for the screen, we had about 250 genes that were derived from a knowledge graph. We used glomerular endothelial cells, a primary human model from the kidney, and wanted to perform high-throughput electroporation to ultimately understand which gene knockouts lead to protection or disease onset. And we used a high-content imaging readout to model a phenotype in chronic kidney disease. I want to focus here in the next 3 slides on how we managed to establish a CRISPR editing workflow in those cells, because I think this is really the part that might be beneficial for those of you on the webinar today that are thinking about an arrayed CRISPR screen in a complex model, but are needing some inspiration on how to edit their model. And the workflows that we've established could be very much transferable to any kind of primary model or even organoids. So when we started the optimization of our gene editing protocol, we had received a cell model that had never been edited before. All we knew was that it was not amenable to lipid-based transfection, so we wanted to use electroporation to deliver our CRISPR reagents. As a first step, we tested different electroporation programs, just simply with a GFP plasmid. All we needed for that was the electroporation kit that we used from Lonza, which came with the electroporation buffer and with a GFP plasmid. We resuspended our cells in that buffer and added the plasmid into our nucleofection cassette and then performed the electroporation. We plated the cells out into our assay plate and then simply read out the percent of GFP positive cells and viability. In a bit more detail, so there was quite a number of programs that we tested with the GFP plasmid. We used a P3 buffer, which is recommended for primary cells. And the programs were recommended from speaking with the Lonza rep at that time. The readout was simply microscopy. I'm sure you can do flow cytometry readouts as well if that's more suitable for your model. And in the end, we simply looked for GFP expression as a readout for efficiency for the electroporation program and a nuclear count to read out toxicity, and chose the programs highlighted here in blue, which were the least toxic and with the highest transfection efficiency. These two programs were then moved into the next step, which were testing the editing efficiency using the guide RNA in Cas9 as a ribonucleoprotein complex. For this step, we needed a few more reagents. So first of all, guide RNAs. Recommend 1 or 2 guide RNAs that are validated, and that by that I mean a guide RNA that has successfully been tested in another model. Either you find that through publications or through recommendations from vendors or through previous projects that you've performed. Cas9 protein that is commercially available, and optional is electroporation enhancer, which I added to the mix in that case. We then used the cells again in the electroporation buffer, the P3 buffer, and manually pipetted this into the 384-well cassette, which we used because we wanted to ultimately scale this up for screen. But of course, for this experiment, we only needed to use a few wells of the cassette. Again, same procedure as before. Cells were transferred in the assay plate, and then we had 2 different options for readouts, either gene editing analysis on the DNA level using sequencing approaches or a phenotypic readout. And in this case, for the renal cells, we did both. We first did a simple Sanger sequencing approach, followed by tight analysis, where we simply compared the sequencing trace between the edited and unedited cells, and we indeed saw a really high percentage of indel formation, indicating very good editing efficiencies. We then also confirmed this with a phenotypic approach. In this case, we added transferrin, fluorescently labeled transferrin to the cells, and if the cells take up the transferrin, they are labeled with red endolysosomes. However, if the transferrin receptor, TFRC, is knocked out sufficiently, like shown here in the middle, the cells are not able to take up the transferrin and are not red. And in this case, again, we could confirm more than 95% editing efficiency. And with a random other guide RNA, in this case P53, we did not see the same effect, so indicating indeed we knocked out the receptor efficiently. So I think I've shown you that in 2 simple steps, we can establish an efficient gene editing protocol using electroporation. And the protocol that has been established can be easily transferred between other Lonza nucleofection devices. For example, if you only have access to a 20 microliter nucleo cuvette, these conditions can then also be transferred to the 384-well nucleofector or 96-well nucleofector that we have here in the FGSL. After this initial setup of the electroporation conditions, there is more optimization that can be done. More guide RNAs can be tested. The amount of the reagents can be titrated and optimized. You can test your conditions with and without electroporation enhancer and so on. But the key step that we had to do in this example for the array CRISPR screen was upscaling the electroporation, because in step 1 and 2, we manually pipetted and only used a few wells, but we really wanted to have a workflow that is feasible for a 384 well format and for a couple of 100 genes at the same time. And to cut a long optimization process short, we have managed to successfully implement a workflow that allows us to edit up to 250 genes at the same time in one workflow in one cassette. This workflow minimizes the seeding variability from the electroporation cassette into the final assay plates, which is really important if you have a phenotypic readout that is depending on cell numbers. And most importantly, this protocol is adaptable for other primary cell models. It has been published, so if you're interested in understanding more about this workflow, please have a look at this SLAS discovery publication from last year. I will move on in the project, and after establishing the gene editing protocol, there is a step of establishing a screenable assay. I will not go into too much details here, just saying that we managed to achieve this. We achieved a really good assay window that was sufficient for CRISPR screening. To be able to do so, we deployed the AI machine learning image analysis tools to help us dissect an untreated from a stress phenotype that you can see here. It's really easily visible by eye, but when we tried conventional image analysis tools that dissect the cells and then measure the intensity at the plasma membrane, we couldn't get such a clean distinction between the two phenotypes that we can see here on the right with the machine learning-based image analysis. Once we had established this screenable imaging assay, we went into the screen and screened 250 genes, both in a under stress conditions and in untreated conditions, and identified indeed hits that either prevented the onset of a stress phenotype in the presence of cytokines IL-1 beta, or that led to a stress-like phenotype in the absence in a healthy state, in the untreated state. And those hits then can be further validated in orthogonal assays and be further characterized in other renal disease models. Just wanted to show you basically how such a workflow for an array CRISPR screening looks like, and particularly focus on how we established a gene editing protocol. I hope that you've appreciated that electroporation helped us to overcome challenges that we have when screening in primary models, and that we were able to scale this up and perform gene editing at scale. And as I mentioned earlier, the FGSL aims to use similar workflows using non-viral delivery of guide RNAs to be able to CRISPR screen in a variety of complex cell models and also organoids. There's a link to the paper and the method below, and I think this is a good time to start a poll and hear from the audience which delivery tools and technologies you are using for your CRISPR screening approaches. And then I will move into the last section of our webinar presentation, where we will focus on the remaining 3 cornerstones of the FGSL: automation, phenotypic readouts, and importantly, the collaboration. So to be able to develop and deliver these arrayed CRISPR screens at scale, we have an automation platform here at the FGSL that's currently being set up. One of these platforms is a dosing platform, which enables us to automate workflows such as guide RNA and Cas9 dispense, compound dosing, electroporation, antibody staining, and also enables incubation steps in between, and many things more. All the equipment on that platform here is accessible in a standalone option, so we can use them for individual experiments in a non-automated way. This entire platform is fully enclosed and enables product protection, which means that we can work under sterile conditions within this enclosure. I wanted to show you quickly what kind of workflows we're planning to do on this platform. As already mentioned, the guide RNA Cas9 dosing and electroporation is one thing that we could run on the platform, as we have all the necessary equipment for Cas9 dosing, cell dispense, moving the cell suspension into electroporation cassette, electroporation, and then also downstream incubations. All of those equipments are hosted on the platform. Another workflow that is more simplistic and very commonly used in our protocols is antibody staining for imaging. For this, we have a wash dispenser that can wash our plates at scale and throughput. We can then dispense antibodies, incubate at room temperature, and repeat this entire circle with a secondary, and ultimately store the plates at a Steri-Store at 4 degrees. So this simple workflow enables us to do high throughput staining of dozens to hundreds of plates in an automated fashion. A second platform that we're building here at the FGSL is our feeder reader platform that enables to automatically feed plates into an endpoint reader. And it also enables incubation from 4 to 37 degrees. The automated endpoint readers that we have is a high content confocal microscope, the Yokogawa CV8000. And the other reader is a high throughput flow cytometer, the Sartorius IQ. In that way, we can run plates and assays overnight and over the weekend. Other endpoint readers that we have that are not integrated in that platform is a Sartorius IncuSite that enables us to lifestyle image. And we also have a plate reader enabling fluorescent and luminescent endpoints, the Envision. There is a lot more equipment that we have installed in our class 2 hoods to enable high throughput handling of cells in a class 2 environment. We're not going into more details of this, but you get the idea. We can do quite a few different endpoints and do this all at scale. Finally, to the last and most important point, collaboration. All of our projects at the FGSL are collaborative projects. We do them either with academics or we can do them with small, medium-sized enterprises. So on the left-hand side, the academic collaborations. Projects are proposed by academics in the UK, and if the project proposal aligns to areas of interest to AstraZeneca, collaboration can be formed between the academic, the FGSL, and AstraZeneca. In that case, funding will be provided by AstraZeneca. But we are also onboarding projects that are not aligned to AstraZeneca's interest. In that case, the collaboration is only formed between the academic and the FGSL. And funding for those projects can be provided by the MRC. I wanted to highlight that those projects, academic projects, are fully funded. That means all the reagents required for an arrayed CRISPR screen fully covered by funds available from AstraZeneca or the MRC, as well as the FTE. So the screens will be executed by the employees here at the FGSL. So really, if you have an interesting idea, this would be a great place to gain access to a platform that most universities don't have. I also want to mention small, medium-sized enterprises or even pharma that can gain access to our screening platforms. This, however, is as a fee for service. And in this case, of course, AstraZeneca would not see the data. There will be a collaboration agreement only between the FGSL and the company. And to keep confidentiality, the analysis can be performed by the applying company, so they fully own their data. Now, a bit more details on how to collaborate with us. The first step is an application form that needs to be filled in. We have a call for proposals constantly open, and the application form can be found here on the Connect Health Tech website that hosts a whole section with guidelines for applications and an FAQ section. This application form that you find there as well is a short questionnaire about your cell model, the cellular assay, and the gene editing strategy that you want to perform in your proposed screen. If you filled in the application form, submit it to this email address, and then we will review it twice a year, in May and November. We will review based on biological relevance of your proposal, but most importantly on the technical feasibility. Applicants will then receive feedback, and successful applicants will then have fully funded screens executed at the FGSL, funding provided either by AstraZeneca or the MRC, as mentioned before. Before the screens, however, are executed, there is a step of generating or agreeing on a collaboration agreement. We work together with the applicant and write a work package, which includes all the deliverables and all the milestones on the way to the CRISPR screen. And again, to mention, the data of this project will be owned by the applicant. One final recommendation for a successful FGSL proposal. So successful proposals will have thought about their phenotypic assay, and the endpoints need to be compatible with the FGSL setup, so imaging, flow, and plate readers that we have here. And the assay needs to be robust, and meaning there's a good separation between your positive and negative control. You need to have thought about a gene editing strategy and deliver some proof of concept data that the gene editing strategy works in your model. And I've shown you today one way of how you can achieve gene editing in complex models. And finally, the proposal is coming, of course, with a biological question and a physiologically relevant model. So we're focusing outside of cancer lines on complex models such as co-cultures, organoids, primary cells, and iPS-derived cells. The FGSL itself is a screening lab, so we are focusing on the scaling up screening and then the downstream analysis of the screening data. I think this leaves me to summarize this last section. I hope I showed you that the FGSL really represents a unique opportunity to collaborate in the arrayed CRISPR screening field. We have access for academics as well as for small and medium-sized enterprises to an arrayed CRISPR screening platform at scale. Successful proposals that come to the FGSL will have tested a gene editing strategy, have thought about a cellular assay, and also their model, of course. And then we can scale up and screen here at the FGSL. This leaves me to show you the FGSL team that we currently have. We're a small team, but continuously growing and currently recruiting more scientists. So also if anyone is interested or knows someone that is interested, please have a look on our website through this QR code. And there's a large number of people that are behind the FGSL, supporting from all the three collaborating parties, the Milner Therapeutics Institute, AstraZeneca, and also the MRC. So huge thank you to everyone involved in setting the FGSL up, in the data and studies we showed today. And that leaves me to thank you all for your attention.
Fantastic. Well, thank you so much, Uli and Wong, for the fantastic presentations. Alrighty, moving right on to the Q&A. Okay, so first question here, one about working with the FGSL. What would a successful proposal look like?
Yeah, a successful proposal for the FGSL will have a good gene editing strategy, ideally non-viral. This could be the two examples that we share, either nucleofection or mRNA synthetic liposuction. The ideal scenario is that you have got a good editing efficiency into your cell model, but also with low toxicity, as well as a good assay endpoint, either imaging or a flow assay that has a good separation between the positive and negative control, so that we can confidently call hits out of those. And a good model that is outside of oncology. Yep.
Excellent. Thanks a lot, Wong. All right. How many screens does the FGSL perform per year, and who owns the data for any joint projects?
Mm-hmm. So At the moment, we're just starting off with our first project, and the next year, 2025, we will perform around 3 screens. So 2 have been decided already for previous call for proposals, and we can have capacity to onboard a 3rd screen, and that will increase in capacity once we're fully operational in 2025. So the years beyond, we will do maybe 5, 6 or more screens. The data is owned by the applicant. So in the case of a joint AstraZeneca and academic collaboration, the academic owns the data. AstraZeneca, however, has a right of research to perform follow-up studies on the data. And in case of the small, medium-sized enterprises, the data is fully owned by the small, medium-sized enterprises. No one else will be able to see the data.
Excellent. Thanks a lot. All right. Can the 384 well and 96 well?
Oh, we stopped hearing you.
I think we might have lost
And 96 well nucleofactor systems be fully automated or integrated into liquid handling systems?
So the question was if 96 and 384 well liquid electroporators can be integrated into automation. And the answer is yes, that can be done. We've done that in our example. We used the Bravo to pipette the solution in and out of the electroporation cassettes. There's certainly other liquid handlers that can do the same. It's probably best to contact Lonza if you want to have a different liquid handler to use for the nucleofection cassettes, or your liquid handling device supplier. And also have a look at the paper that we mentioned earlier that shows the workflow, how we used semi-automated workflows on the cassettes using the Bravo.
Excellent. All right, great answer. Okay, in your experience, to what extent will using relevant cellular models improve target identification processes and drug discovery in general? And can you share any examples?
Shall I go again? Yeah. I think, yes, we highlighted earlier, we're really keen on using disease-relevant translatable models because the translatability and the success of targets that come from translatable models in the clinic is just much greater. I think there's papers and studies showing that the reason why drugs fail in the clinic is efficacy, and models often don't translate in the drug discovery process into efficacy and then further down in the patient. So that's the reason behind complex models. They just, if you characterize your model and compare it to tissue organ-derived data sets, they overlap better than a cancer cell line with a tissue profile. So that's really why we want to use complex models. I think there might be lots of examples in the clinic or in the not in the clinic, basically failed examples where there was no translatability of studies in simple models, basically.
Yeah, great points. All right, interesting question here. So for transfection optimization, how many hours after transfection did you perform the GFP cell quantification? And would you be able to tell us the program that worked for you?
So I did look into GFP expression only 24 hours after, because it should be quite quick to be expressed. I have the program not at the top of my head, but that can be shared. And I think the whole study will be wrapped up and hopefully come out as a preprint in early 2025. So yeah, it can be shared.
Excellent. All right, so how did you select the different electroporation protocols you tested with GFP to optimize for gene editing?
So I did reach out to Lonza, to one of their technical experts at the time, Peter in Cologne. I've heard he's still there, who helped me select, basically recommend a number of programs to test with. I think there's also a matrix of basically, usually there's a lot of expertise in Lonza on which cell type to use in which program, and then use a variety of programs that are either more efficient or more toxic. So there's a range of programs that can be tested. So in this case, I tested 15 programs. It sounds a lot, but I really had no starting point in terms of there was no literature evidence of this glomerular endothelial cells being edited. So I tested quite a wide range. And the reason why this was tested with a GFP plasmid is this is really cheap. So you want to test a wide range of programs. The cost is only this little bit of buffer into your cells. If you test 15 programs with the guide RNA and Cas9, that's much more expensive because those reagents are expensive. So I would definitely recommend, if you have no starting point, reach out to Lonza. They will help you to find a starting point, and then test your first variety of programs with a GFP plasmid that is anyways delivered with your electroporation kit. And that worked really well in my case. I had actually 2 programs that worked almost identically well, and I just selected one that was a tiny bit better. And yeah, as I mentioned, that can be shared and will be shared in the preprint next year, the latest.
Fantastic. Yeah, nice little shout out to Lonza, who sponsored this event. So yeah, thanks, Lonza. All right, so somebody here said that they're interested in performing the skeletal muscle C2, C12 cell lines for the CRISPR-Cas9 model. They ask if they can apply to FGSL, and maybe you could just remind the audience here how people can apply to work with you.
Yeah, I think the application is quite straightforward. So you can submit a model or your assays to, we have got all our forms and guidance on the Connect Health Tech platform. And you can access this through the link to our website, which is in the handout. So it's quite straightforward. It's a kind of a survey to just fill in some details. We have got a biannual project calls for project proposals that we review them. And I think from memory, C2, C12 is murine cell line. One key thing is, if I'm not mistaken, so we can only do human cell models, basically because of restriction to our CRISPR libraries. Yeah.
Our libraries only target the human genome.
Mm-hmm.
And another thing to mention, if you're interested in submitting an application, please feel free to reach out to us. We're happy to have a chat and also help you with, if you still have some questions around what the best assay, what the best editing methodology is. We're happy to help with that and get your proposal and your model ready for screening.
Excellent. Yeah, thanks a lot. Okay, so is the Functional Genomics Center at the Milner Institute an extension of the Cancer Research Horizons at Cambridge? They're saying both. They're guessing both centers are collaborating with AstraZeneca.
I guess I can take this. We are not an extension of that. So that's a different kind of initiative driven by the Cancer Research Horizon with AstraZeneca. So that's the main overarching goal of that is to perform pool CRISPR screens in cancer models. And we are not an extension of that. We are a separate kind of a center that's driving array screens in non-oncology model. We do have close contact, but we are not an extension.
Well, I guess the success of the Functional Genomics Center inspired the start of a new collaboration to address this gap of arrayed CRISPR screens in non-oncology models that we're trying to address here, and giving people access to arrayed CRISPR platforms and automation that most universities don't have access to. So separate things, not as famous as the Functional Genomics Center yet, but hopefully in a few years' time, people know we're separate places and do separate things.
Yeah, definitely. All right, so what are the major driving costs of the CRISPR screen?
There is a couple of costs, I think. Let me think from the top of my head. If we do electroporation that is not cheap. I think, first of all, bespoke guide RNA libraries, synthetic guide RNA libraries come with a cost. If we do electroporation of guide RNA and Cas9 and RNP, the protein, Cas9 protein comes with a cost. The model itself often comes with a cost. So particularly if we look more into 3D cultures and bespoke plates and media that the cellular cultured in, they also come with a cost. I think there's, depending on the model, different components. I think the more complex the model, the more bespoke the culture conditions and the assay plates also are. So yeah, basically guide RNAs, Cas9, and cell culture. Of course, if you start from scratch, I mean, all the equipment you might need to automate and scale up the process also comes with a cost, but it is just the infrastructure basically, and that would be provided by us, and also the costs would be covered by successful proposals that we onboard here. So it's more something for us to think about. If you are interested in working with us, you don't need to worry about the cost too much because that will be covered.
Excellent. Yeah, no, that's great to hear. All right, I think we're running out of time and questions, but maybe we'll just end with one. So in this field, can you see any developments or exciting research opportunities that you're especially looking forward to over the next couple of years?
Yeah, I think that's a good question. I think in the field of screening, I think there's a lot of CRISPR screens that's been done in cancer models in a more high throughput manner. I think one key thing that really excites me is to actually apply this in non-oncology models, models that have not really been edited before, as one of the key examples that Uli showed was in renal cells that have not actually seen CRISPR-Cas9 reagents before. And I think that's something that really excites me, that we can find new biology that really drives disease progressions that really understudied and really we can really find new targets out of it that can help patients.
Fantastic. And Uli, what about you?
I think what is exciting is also the application of more machine learning based tools, be able to multiplex more assays. So with the limit of available cells in these complex models that we are aiming to work with, we really want to make most of the endpoints or of the screen that we get out to. So the Wong showed a really nice example at the beginning, how different endpoints can be multiplexed in an imaging assay. You can even go a step further and take the supernatant and look at the supernatant and then perform an imaging endpoint, or have duplications and have completely different readouts. I think this possibility to multiplex and be able to handle large data sets, this is something definitely we want to look into, and we would be really excited to see more.
Excellent. Well, yeah, thanks so much, Uli and Wong, for the great presentations and insights in the Q&A. It's really been a pleasure having you with us.
Thank you.
Thank you very much.
Yeah, big thanks also to the audience for participating in the webinar here. And in closing, we hope you enjoyed this Scientist.com and Lonza webinar, and we'll see you again next time. Have a great day, everyone.